Okay, so let's just review that just briefly. Remember that we said skeletal muscle tissue is highly innervated, and every single skeletal muscle fiber must have direct innervation, or else it's going to be paralyzed, right? The site at which a motor neuron innervates a skeletal muscle fiber is called the neuromuscular junction. And you're not going to be responsible for this now, but I'm going to give you a preview of what you're going to learn in physiology. Okay? So basically at the neuromuscular junction, what you're going to have is this right here is the end of the motor neuron that we were looking at, like the cell body would be up there somewhere. And then this area right here is actually called the synapse or the neuromuscular junction. And when you get to physiology, you will learn that in excruciating detail, what's actually going on there in excruciating detail. Basically, it's a chemical communication between the two of them. And then in phys, you learn how that's related to tetanus or muscular dystrophy or all of that kind of stuff. So what we had to get you guys to understand in anatomy is just this is what this guy is, and then you'll learn the actual physiology of it when you take phys. Okay, so, and then the last thing that I told you guys last time, I think, was that each individual skeletal muscle, in addition to being highly innervated, each fiber was also highly vascular, right? Every little fiber had like a sweater of capillaries all the way around it. Okay, so what we're going to do now is we are going to go down and look at the microscopic and submicroscopic nature of skeletal muscle. Why the hell do we need to do this? The reason we need to do this is because this corresponds to understanding muscle functionality. You cannot actually understand what's going on to get you, for instance, to pick up a water bottle, right, without going all the way down to the submicroscopic level of an individual muscle cell. So the stuff we're doing now has directly to do with phys and less to do with lab, okay? And it also has to do with really functionally understanding these things. So what we're going to do is we're going to take it and we're going to break it all the way down to the submicroscopic level so we can get the functionality, and then we'll bring it all the way back up to the gross anatomy so you can see how that is related to muscle movement. Okay? All right, so let's start with what we know about skeletal muscle fibers to begin with. Remember that when we start talking about a skeletal muscle fiber, what we are talking about is the entire muscle, right, is a bundle of bundles, right? And each individual bundle is called a what? Bundle of muscle fibers is called a what? Fascicle, right? And then if you break down the individual fascicles, you will see that each fascicle is composed of a bundle of muscle cells, also called muscle fibers. But please note that if this is a cell, it doesn't look a damn thing like the cells you looked at in 1610, does it? Right? However, it will have some things in common. It will have a cytoplasm, cell membrane, most of the same organelles. Then it's got all of these unique structures that you've never seen before. These unique structures are related to its unique function, which is contraction, right? So it's going to be basically a cell plus contractile ability. And so what we're going to get to today is how on earth do those structures relate to contraction, and what do they do to the structure and the function of the cell. Okay, so let's go back to what we know about skeletal muscle fibers or cells. So here's one skeletal muscle fiber right here, or cell. I know it looks weird. Let's blow it up a little bit more and make it transparent so that you can see it. So what we are actually seeing right here is a skeletal muscle fiber or a cell. Notice that it does have some things that you've heard of before in it. It's got some mitochondria, it's got some nuclei, right? It's got some other stuff in it. But unfortunately, some yahoo decided that they were going to rename every cellular structure after you just learned the structure and function of them in 1610. Why did they do it? I don't know. They hate us all. They did it to me too. It was long before I even took this stuff. So what they did is, instead of, for instance, calling it cytoplasm, which you were used to, it's now sarcoplasm, right? Instead of calling this the cell membrane, and you know about cell membranes, right? You even know what they're made of. What are they made of? Phospholipids with proteins embedded in them. Same damn thing, only now you're going to call it the sarcolemma. Sorry. Why am I making you learn these terms if they're arbitrary? Because that is how they refer to them all going through the rest of your career. So you actually have to know the terminology. I'm just trying to put it together with the fact that it's not different than something you knew before. It's just a different way to refer to it. So they renamed everything, almost every damn thing in there. So what we are going to do is we're going to look at what it's similar to the cells that you've already met and how it's different from the cells that you already met. First thing to acknowledge is that this is an actual cell. That's the sarcolemma, which is the cell membrane. Somebody think about this question and answer it for me. What is the relationship of the sarcolemma and the endomysium. How are those structurally related to one another? Other way around. So the endomysium would be superficial to the sarcolemma, right? Right, so you run into cell structures underneath the endo. Does that make sense to you? Okay, so the endomysium is actually a separate tissue that's wrapping around each individual cell. Does that make sense? So it's superficial. Or you can say the sarcolemma is deep to the endomysium, right? But once we get to this level, we're actually at the cellular level. We're actually looking at the cell. Okay, the other thing that you know and have known is that skeletal muscle tissue is striated. So I want you to begin to see the nature of the striations by looking at an individual cell, and then we'll extrapolate up to the whole tissue. And so what you will see is if you start looking at this individual cell, do you see this region where it's slightly lighter, and then this region where it's darker, and then slightly lighter again? You're going to do that repetitively all the way down the cell, right? And then the cells are going to be lined up so that the light lines up with the light and the dark lines up with the dark, which ends up making the entire tissue look striated, okay? And then the other thing that you know about skeletal muscle from even histology is that skeletal muscle fibers tend to be multinucleated, yes? And the nuclei are peripherally located. So nucleus, 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 nucleus up there, nucleus there. Can you now start to conceptualize why the nuclei are peripherally located? Because isn't the middle of the cell kind of packed, right? So they get smashed up against the side because the middle of the cell is completely pretty much dedicated to contraction, and we'll talk about the contractile nature of it. Does that make sense? So multinucleated, peripherally located for reasons you can understand now. Okay, so now let's look at some other things, similarities and differences from other cells. First off, this is longer than most cells you're going to run into. A single skeletal muscle fiber or cell is up to a foot long, but it's microscopically thin, 10 to 100 micrometers. And the micrometers, the you generally have to use a microscope to see, right? So like fishing line, but thinner than fishing line, okay? And in me, 12 inches, you wouldn't be at all surprised about. And Shaquille O'Neal, they get longer than 12 inches, right? Way the hell longer, right? Okay, so we had 12 inches of each individual. They can be up to 12 inches long. Like, for instance, you know the longest muscle in your body? The Sartorius. The Sartorius wraps from here all the way around. You are going to find your longest muscle fibers in the Sartorius. And in someone as tall as Shaq, they're going to be incredibly long, very long muscle fibers. Okay, now let's start looking at internal structures and similarities and differences. So of course it has a cell membrane, because you can't be a cell, whether it's eukaryotic or prokaryotic or plant or animal. You can't be a cell without a cell membrane. So of course it has a cell membrane. Cell membrane has all the crap you expect it to have. It's made of phospholipids. It's got proteins embedded in it and all that business. But you're going to call it the sarcolemma, okay? I don't know why they decided that, but the sarcolemma. It's actually called the neurolemma in neurons, so at least after you learn it here, it'll like work for them too. And then inside, of course, you are going to have sarcoplasm. Same thing as cytoplasm. Now, notice that there's not a whole hell of a lot of room for sarcoplasm because it's so chock full of everything, right? But there has to be sarcoplasm wherever this is like internal spaces and no organelles, right? So what's in the sarcoplasm is some stuff that you expect and some stuff that you might not have expected. Of course, you know, the nuclei are in the sarcoplasm, and then the mitochondria are in the sarcoplasm, and a bunch of other organelles. FYI, skeletal muscle compared to other muscle or other tissues tends to have way more mitochondria per cell. Anybody put that together with me? Why? Why would you have way more mitochondria in skeletal muscle fibers than in other cells? Go ahead.
Because they use a lot more energy.
Yeah, it's incredibly energetically expensive. I'll sort of get you guys to start to conceptualize how incredibly energetically expensive skeletal muscle contraction is today. But they have a lot more mitochondria. So you're going to see a whole bunch of mitochondria. You're going to be multinucleated. You're going to see some other structures as well. But then there's a couple of things in the sarcoplasm which aren't visible because they're macromolecules and not organelles. But they're important to understand the structure-function relationship. So let's talk about them. There's a couple of things that will be dissolved in that sarcoplasm that are functionally important. One of them is that in the sarcoplasm of skeletal muscle fibers, there is glycogen stored. And glycogen is stored what? Stored glucose. Okay, so keep that in mind. We're developing a pattern here. We got boatloads of mitochondria and we've got stored glucose. right? In addition to the glycogen that's stored in there, you also have a protein called myoglobin. And myoglobin is similar to hemoglobin. Hemoglobin binds to and carries what around in the bloodstream? Oxygen. Myoglobin binds to and stores small quantities of oxygen inside each skeletal muscle fiber. So now what I have set up, guys, and I hope you remember this much 1610, or I'm going to tell Kristen if you didn't, okay? Tell all your 1610 professors that you didn't remember it. So now what I have is glucose stored as glycogen, yes, inside every single fiber, and I have oxygen stored on myoglobin, and I have a whole bunch of the locations that most of cellular respiration occurs in where? Mitochondria, right? So what I have is every single skeletal muscle fiber is set up to make sure that even if you haven't eaten in 36 hours, you can get out of the way of the damn bus, right? So it's always got a piggy bank. And you're thinking, well, some of you guys who know that glycogen is stored in the liver, you're like, oh, well, glycogen is stored in the liver. That doesn't get you out of the way of the bus. You need it faster than that, don't you? Right? So you need quick access. So everyone, instead of waiting for the liver to send them glucose, has its own little piggy bank that it always keeps stored, right? So that you can always do a little bit of muscle contraction, a little bit of emergency response. This is your emergency response tissue, and it can always generate a little bit of ATP for contraction, right? Now, you're going to peter it out really quickly, but hopefully the bus is gone by then, right? And you're going to eat something too. Okay, so now, the other thing about myoglobin is that myoglobin is similar to hemoglobin in another way. When oxygen binds to hemoglobin, what does it do to the color of your blood? Makes it redder. Yeah. My son, who is 11 years old, will still not believe me that your blood is not blue. And I'm like, it's literally not blue. And he's like, I don't believe you. I'm like, who the hell would you believe? Like, I cut up people all the time. There is no blue blood. But no, your blood is not blue. Your blood is red and redder. Okay? It looks blue because it's actually being sort of, you're seeing it through the pigments of your skin. When it has a lot of oxygen, it's redder, right? The same thing inside this, since there is myoglobin inside there, when the myoglobin binds to oxygen, that is the other reason that muscle tissue is red. So that cross-section of a cow muscle called a steak, right, now is red because every single cell has a little red sweater on, right? And then the tissue, the cell itself is actually red, right? That makes sense? What happens if you take that cross-section of the cow muscle and leave it out on the counter for a long time? It becomes brown as the oxygen dissipates out of the hemoglobin from the capillaries and out of the myoglobin from the cells, right? How can you heat up that process or speed up that process? You can just heat it, and then you get rid of the oxygen really quickly. That make sense? Okay, so now let's address the elephant in the room. The hell are these things, right? Big old Vienna sausage looking things that nobody taught you about in 1610, right? These things are big bundles of contractile proteins that attach to the cytoskeleton at either end of the cell. And those are going to be the things that contract. And when they contract, and I'll teach the mechanism of contraction today, or at least a short version of the mechanism of contraction today, when they contract, if they're attached to the cytoskeleton, what's going to happen to the length of the cell? You're going to shorten it. If you contract enough of the muscle fibers in a fascicle, you can shorten a whole fascicle. If you contract enough of the fascicles, you can actually pull on the tendons. If you generate enough tensile strength by pulling on the tendons, you can pick up a water bottle. Yeah? You have to overcome the weight of the bone and then of anything else that's on there. You have to be able to generate enough tensile strength to overcome that. Does that make sense? Now, briefly, and we talk about this in a lot of detail in phys, if I could make this have more contractile proteins, if I could do some type of activity that would increase protein synthesis of the contractile proteins inside each muscle cell, if I could make it have more contractile proteins in each of the muscle cells, can you see that I could probably generate more tensile strength? Yeah? So if your muscles are bigger, yes, you have bigger muscles, you actually have potentially stronger contractions because the nature of the contractions is in these contractile proteins. Does that make sense? So we're going to talk about how the contractile proteins result in things shortening in just a second. You guys all right with that? Go ahead.
So if you have bigger muscles, does it take more effort to contract?
Bigger muscles, if you have a lot of muscle mass, first off, after muscle tissue is generated, it's never completely relaxed. It always has a partial degree of tone. So if you're just sitting around and you're like really muscular, you're burning more calories than someone who's sitting around and it's just kind of flabby, right? And yes, so sitting around as a muscular person requires more energy, right? And then when you actually lift things as a muscular person, it also requires more energy. So it actually ends up burning more calories. It's energetically expensive to be very muscular, right? Which is why the metabolism of people who have put a lot of work in on the front end, right? Getting all muscly, they just get to sit around and like eat a cheeseburger and feel superior to all of us, right? But it's because they paid for it already. That's what they did, right? Or their genetics paid for it, and that's what pisses me off. You're like, just genetically muscular. God dang it. Okay, so now let's talk about those big Vienna sausage things. These things right here is a cable-like bundle. So think of a coaxial cable. Do you know what those are? It's like a cable made of cables, right? A wire made of wires. Yeah? Coaxial cable?
Table made of what?
A cable made of cables or a wire made of wires?
They said a table made out of tables.
No. You're like, wait a minute. This is getting weird. Are we talking philosophy? Okay, a cable made of cables, right? So what this thing is called a myofibril, and the myofibril is actually made of smaller things. These are all proteins, guys, so let's put it back together with cell biology for a second. I know you guys are like, I forgot about cell biology. I thought we were doing more fun crap. No, we're going back for a minute, okay? These proteins are long, skinny proteins that would have been made where inside this cell? You can do it. Long, skinny proteins that would have been made where inside this cell? Proteins, yes, say it. Ribosomes. Ribosomes, yes. The ribosomes would be making these proteins inside the cell. So they've got these long, skinny proteins. So this myofibril right here is going to be the basis of the striations. It's also going to be the basis of contraction. So what we're going to do, we're going to take one of these and break them down and look at it two-dimensionally so that we can sort of get it simplified. But what I want you to see before I start drawing it two-dimensionally is what we're going to look at is this repeating pattern. Do you see this jagged line? Yeah? Then a light band, then a dark band, then another light band, and then you hit the jagged line again, right? That's going to repeat over and over and over again down the length of the myofibril, like boxcars on a train, right? Over and over and over and over again. So what we're going to do, we're going to take one of those, we're going to flatten it out 2D so it's easier to see, and then we're going to build it on the board so that you guys can understand it. And then this will, by the time we're done, we will understand both the nature of the striations and also the preview of the contraction mechanism. Okay? You guys ready? All right. So this has more detail than is really important in it, so I'm going to draw the really relevant parts on the board. But what I want you to see is this. Do you see that jagged line? We noticed that before, right? And then we are going to see this jagged line. That's the edges of the repeating unit. Now, if what I do is use a protein staining technique, then more protein will stain darker and less protein will stain lighter. Does that make sense to you? Okay, so let's start building this and then we'll get the pattern down. All right. So what I'm going to do is I'm building one boxcar, right? So I've got this jagged line. From one jagged line to another is one boxcar called a sarcomere. And multiple sarcomeres will make up that myofibril, the Vienna sausage thing. Yeah? Okay. So what I'm going to do is I'm going to try to use the same colors that they do. I'm not going to build every little thing in there because some of them are structurally important, but they're not functionally important. Okay? So let's look at the important ones. Notice that big fat protein in the middle with all the things sticking off of it. That one is going to float in the middle. It actually doesn't float. You can see that there's something holding on to it. But this protein is the thicker of the two. Its name is myosin. and they call it the thick myofilament. What does myofilament mean? Muscle, yeah, basically a long, skinny muscle thing. What? Long, skinny muscle protein thing is what it is. Does that make sense? Okay. So you see it in there. Now, it's got little projections sticking off of it that are going to be important. So I'm going to go ahead and draw them in now. And those little projections are called Myosin Heads. Okay, so we got the myosin. Now the red ones, notice these red guys right here that are attaching to the Z lines. Sometimes, by the way, they call these Z discs. That's fine. Z lines is more common. That's what I'm used to. Z discs is also fine. So the red ones actually attach to the Z lines and go toward the middle. So we're going to draw those in. Okay, that is a different protein. It doesn't quite touch in the middle. So this is called the actin thin myofilament. And before we get to functionality, let's look at how this can result in striations. Okay, which protein is thicker, actin or myosin? Myosin. So if I use a protein staining technique, what I will get is wherever myosin is, it will be darker than where actin is by itself, correct? Okay, so what you're going to end up with is wherever myosin is going to be the darkest part of the tissue. Does that make sense? Okay, so this from here to here is called the A band. A stands for anisotropic, which you don't care about, but it's also the second letter of dark. D-A-R-K, so just keep that in mind. So let's find it here. Do you see the A band from here to here? Yeah? Notice that in the middle of the A band, there's a slightly lighter area. It's not as light as out here, but it's slightly lighter. Why would it be lighter? Well, we're using protein staining, right? And this one has more protein because it's got actin and myosin on top of one another. This one has slightly less, but still a lot of proteins. Does that make sense? So this portion right in the middle, from here to here, is called the H zone. It's a little lighter, and it's in the middle of the A band. And then what I'm going to get from here to the next myosin, which isn't drawn here, from here to the next myosin is going to be the lightest part, and that's called the I band. So do you see the I band right here? Okay, so now let's talk about how this results in the tissue being striated. Okay, so what I have is all along a whole myofibril, these will be lined up like this. And what it's going to look like from far away is basically a light band and then a dark band and then a light band and then a dark band, okay? But if you line all of them up together instead of just one cell being striped, the whole tissue is going to be striped, yeah? So the tissue ends up looking striated. And if you ever looked at the tissue up really close, you could see that it wasn't just light and dark striations. Let me find a picture for you real quick. Let me find a good skeletal muscle. Where are you, muscle? There you are. Okay, so if you look at this figure, can you see that you've got a dark stripe and then a light stripe and then a dark stripe and then a light stripe? But can you see that there's actually even a little lighter part in the middle of the dark stripe, right? What is that? What are you seeing? The little light part in the middle of the darker stripe? Yeah, you're seeing the H zone, yeah? So if you look at it really closely, you can start to see this whole pattern developing right there. So that's what you're looking at. Okay, so what it ends up doing is this structure ends up striping the whole tissue. Now, why the hell do we care? Like, that's a lot to just describe why the hell a tissue is striped. Well, we do care because this ended up giving us an idea about functionality long before we had individual protein staining techniques. I just need to add one more structure, and then I'm going to start telling you how this works. Okay, one more structure is that on the actin, there are actually binding sites for the myosin heads. They don't look like this, but it's the easiest way to draw them. So I'm just going to draw them as like little handles. What they are is actually chemically attractive binding sites, but I'm going to draw them as handles. And they are called the actin active sites. So, actin active sites. And now we can talk about how contraction occurs. Because the basics, although there's a lot more detail that you'll learn in phys, the basics of how contraction occurs is that when the nervous system tells it to, what is going to happen inside this cell is that the myosin heads are going to reach up and grab the actin active sites and pull. And when they do that, what will happen to the distance between the Z lines? It will shrink, exactly. And so if I shrink enough freaking boxcars, I can shrink the whole train, right? Shrink enough sarcomeres, you can shorten the whole myofibril. Shorten enough myofibrils, you can shorten the whole cell, yes? Shorten enough cells, you can shorten the whole fascicle. Shorten enough fascicles, you can yank on the tendon. Does that make sense to you guys? Okay, so this description of this mechanism, we actually came up with before we could do protein staining to see if we were right. And we came, is there still something? Okay. So what we came up with first, when I took physiology when I was in college, it was before PETA, so we just still did animal experimentation even at undergraduate level. So what we would do when we took physiology and we did the muscle physiology, we'd take a frog. I like frogs just as much as the next person. And we would pit the frog so that they can't feel anything. And then we would take their gastrocnemius muscle and stretch it over a light microscope. And then we would zap it with an electrode. And what would happen is looking at it relaxed, it would be striped, the tissue, right? But looking at it contracted, it would get really dark. Because what actually happens is the dark part, the A-band, right? The A-band stays in position, and what happens is the A-band stays in the middle, and it's kind of like you were on the monkey bars as a kid, only you were a badass, and the end of the monkey bar has to come to you. Right? So you were standing right there and you were pulling the end of the monkey bar all the way into you. And you are the A-band, right? Only you are doing it in two directions. So I told you before, it's like the myosin heads reach up and grab the actin handles and pull. But they don't pull in one fell swoop. What they do is ratchet. They reach up and grab, and then grab the next one. So, and they're doing it on both sides. And here is how you can understand why it's so energetically expensive. Because every single requires an ATP. Every single one. It's incredibly energetically expensive tissue, right? Every single one in every single sarcomere, which is 3D, not 2D, right? Yes? And then multiple sarcomeres in each myofibril, and then multiple myofibrils in each cell, and then, I don't know, I'm just going to make up a number again. Let's say 10,000 fibers in a big muscle. Will somebody look that up for me and teach me something? I've been teaching you. Look up how many muscle fibers in a muscle. A small one and a big one, like the glutes. So I want to know how many muscle fibers in glutes. Anyway, a lot. More than 10. Definitely. Okay? A lot of ATP is going to be used. Okay. Does that make sense to you guys? So now, to confirm that, long before we had protein staining techniques that could stain actin and myosin different colors, we could actually confirm that. We thought it was happening. They were like, oh, what looks like it's happening. So look at these three in a row. You ready? Okay, this is a relaxed sarcomere. Look at the length of the A-band and the length of the H-zone and the I-band. This is partially contracted. Notice how much darker the whole damn thing gets. This is completely contracted. This is what I saw with the frog muscle as I was zapping it. Although it didn't have sound effects. Does that make sense to you guys? Okay, so how they describe this is They say that this is called the sliding filament theory of muscle contraction. Please do not be one of those poorly educated people who thinks that something that is a scientific theory is something that we're still arguing about. That is not what theory means, okay? Theories are as far as you get in the life sciences. Laws you get in the physical sciences. It's terminology, and it does not mean that there is not enough evidence. We have actually stained this and seen plenty of evidence. The cell theory, it's not like we don't know that living things are composed of cells, guys, right? The sliding filament theory, the theory of evolution, all have tremendous amounts of evidence for them. So they call it the sliding filament theory because what is actually happening? What are actin and myosin actually doing? They're sliding past one another, right? That's what they're doing. Is actin or myosin, either one of them, actually changing length in the process? No, they're not changing length. They are sliding past one another. Now the sarcomere is changing length, right? And the myofibril is changing length, but each individual filament, myofilament, is not changing length, right? So this is called the sliding filament theory of muscle contraction. When I was in college, they still didn't have the individual protein staining techniques that could stain myosin a different color than actin. So we were pretty damn sure that this was what was going on, and we watched it happening under a microscope. But then after we got individual protein staining techniques, which was when I was in graduate school, we actually got to stain actin and myosin a different color from one another and actually see it happening under the microscope. So this is the sliding filament theory of muscle contraction. There is a lot more to it. You can talk about when things bind and where ATP is used and the physiology of it. But basically the sliding filament theory says that upon stimulation by a somatic motor neuron, actin and myosin filaments will slide past one another. And the Z lines come closer together. And the H zone and the I band will change width. But what about the A band? The A band stays the same, and the filaments don't shorten, but the sarcomere does. Yes, Alex.
Just for the muscle fibers, I don't know if you actually want it.
Yeah, I do want it.
Okay, so it actually depended on the age, but the estimate for younger people was 253,000 muscle fibers in the bicep brachii.
Ooh.
And then in older men, it was 234,000.
Still pretty damn impressive, right?
Bodybuilders have up to 400,000.
Yeah.
Bodybuilders have up to 400,000? Oh my God, I'm going to try to remember that number. I can remember 250, right? $250,000. $250,000 in the biceps brachii. Now I want to know how much in your booty. Gluteus max.
That's according to the U.S. Library of Medicine. So.
Okay, awesome. I love that. I'm terrible at remembering numbers, so I usually just make up something that's like true-ish and then tell you I'm lying. So I'm like, I don't know, it's like this. Okay, you guys ready? Okay, so now what I want to do is I want to take this and make it relevant to whole muscle, right? So what we are going to do together is we are going to go all the way from the whole muscle that you guys understand, all the way down to the submicroscopic level, and then make you guys use the words correctly. Because otherwise you will just memorize this and it won't have anything to do with the actual biceps brachii, correct? So I'm going to do it all the way down and then all the way back up, and you guys are going to do it with me, okay? So let's start with the gross anatomy. A whole muscle, like the biceps brachii, will be covered in connective tissue called what? Epimysium. Yes, good. And a whole muscle will be composed of multiple bundles of muscle fibers. Yeah? A bundle of muscle fibers is called a what? Fascicle. Each fascicle is wrapped in connective tissue called what? Perimysium. Good. And then each fascicle is composed of multiple muscle cells, also called muscle what? Fibers. Yes. Okay. Each individual muscle fiber will be wrapped in connective tissue called what? No, not sarcolemma. Endomysium, sarcolemma, you can't use them interchangeably. Endomysium. Deep to the endomysium connective tissue, you will run into the muscle cell membrane called the what? Sarcolemma, exactly. So you run into the sarcolemma. The sarcolemma contains a bunch of organelles and other stuff, right? And inside the sarcolemma, inside the sarcoplasm of the sarcolemma, are a bunch of cable-like bundles of contractile proteins. One of those cable-like bundles of contractile proteins is called a what? You can. Myofibril, okay? The myofibrils are attached to the cytoskeleton. I can break down the myofibril to individual contractile proteins, a thick one called myosin and a thin one called actin. The striping pattern of the muscle tissue is caused by the pattern of arrangement of actin and myosin. Yeah? Okay. From one Z line to another Z line, that pattern repeats. What's the repeating pattern called? Sarcomere. Exactly. Does that make sense? Okay. Now we're going to go back up. I want to make a sarcomere. Okay. Which of the myofilaments attaches to the Z line? Actin attaches to the Z line, yes. And then the thick filament, what floats in the middle? Myosin floats in the middle. Okay. From one Z line to the next Z line, the repeating pattern is called a what? From one Z line to the next Z line, the repeating pattern is called a sarcomere. Exactly. Take a whole bunch of sarcomeres and line them up like boxcars on a train, right? And you make this long cable-like bundle of contractile proteins. That cable-like bundle of contractile proteins will be attached to the cytoskeleton. It's called the what? Myofibril. It's called a myofibril. Take a whole bunch of myofibrils, put some other organelles and some sarcoplasm in there and wrap it up. You will wrap it up with a cell membrane called a what? Sarcolemma, exactly. And you will call that cell a muscle what? Fiber, exactly. Each muscle fiber is wrapped in connective tissue called what? Endomysium, yes. Then take a whole bundle of those muscle fibers with their endomysium and wrap them together. That bundle of muscle fibers is called a what? No, the bundle of muscle fibers, fascicle. Now wrap that fascicle in connective tissue called what? Perimysium. Now take all of those bundles of muscle fibers and wrap them up and make a muscle. The whole muscle is wrapped in connective tissue called what? Epimysium. Exactly. Does that make sense? So what happens when you start working out? Let's talk about weight-bearing activity, like you are actually lifting weights, okay? Because it's different when you're doing things that are not really straining your muscles dramatically. When you start lifting weights, what you don't get is after embryonic development, you hardly get any more muscle cells at all. As a matter of fact, muscle cells are pretty much amitotic. You don't really get any more. But what you can and do is that you can increase the number of contractile proteins in each myofibril, and you can increase the number of myofibrils in each muscle cell. So what happens? Do you get more muscle cells or do you get bigger muscle cells? You get bigger muscle cells, yes. And that's really only if you do a certain type of weight-bearing activity, a weight-bearing activity that's about 65% of your maximum contractile tension or more. That's why if you're trying to build muscle mass, do you lift something light 25 times or do you lift something heavy 8 times? Yeah, something really heavy 8 times. You're trying to get to about the maximum, close to the maximum of what you can do. Okay, you guys all right? So do you see the relationships between these things? If you memorize this and you don't have any idea what it has to do with whole muscle, then you're not going to do well, right? I need you to put it together to make a story out of it. Go ahead.
So in the definition of the IBED, it says composed of actin, filament, alone.
It is.
I don't really understand the IBED. Isn't it just from the end of one myosin to the next?
Yeah, but it's just got actin in it. Do you see? So right here. So red is the actin. So don't worry about the green because the green is actually just a springy protein. It's not contractile. So what's going to happen is you're going to be in the I band from the end of one myosin to the beginning of the next, which will only have actin in it. Does that make sense? And also the Z disc, but the Z disc is also just a structural and not a contractile protein. So what I had here is I really only taught you the proteins that were involved in the contraction process. There's some other ones that are like space holders and springs and stuff like that. Does that make sense? Okay, all right, so now all we need to do with our last few minutes is use this to compare and contrast between cardiac muscle tissue and smooth muscle tissue. So it probably won't surprise you at all that cardiac muscle tissue has an awful lot in common with skeletal muscle tissue because it has the same banding patterns, right? And it has the same banding patterns because it has the same kind of sarcomeres. It has actin and myosin, and the whole thing is very similar. The whole internal structure. Now what's really different is how you get cardiac muscle started. It's not under voluntary control, right? Do I have to stimulate every single cell in cardiac muscle? No, I don't have to stimulate them. How can I get around that with cardiac muscle? How do you get around having to stimulate every single cell? What? Gap junctions. It's gap junctions, right? So if you guys were all in cardiac muscle and I was the nervous system and I stimulated her, right? She could cause all of you to contract sequentially afterward, right? Because of gap junctions. So I can get around the need for individual innervation. So cardiac muscle is not innervated as many places at all as skeletal muscle. So it is, however, striated. And of course, the cells can communicate with one another at the intercalated discs, where you have your gap junctions and your desmosomes. So it is striated, but it's under involuntary control. It's got the same arrangement of actin and myosin. It's also got lots of mitochondria. One of the problems with cardiac muscle is that it does not store oxygen, though. So you're kind of hosed. If it doesn't store oxygen, and as myoglobin, it doesn't store much, almost none, and you cut off blood supply to it, what happens to cardiac muscle tissue? It dies. It's amitotic too, but it's a much bigger deal. Because skeletal muscle tissue is amitotic, but it's also multinucleated. So if I cut off blood flow to a region and I just lost one nucleus, but I've got 17 more, can't you, you didn't actually kill the whole cell, did you? You only killed a region of the cell, right? But with cardiac muscle, if you kill the whole cell, you're screwed, right? It's gone forever. You'll just replace it with scar tissue. So each fiber does not need to be stimulated by the nervous system to contract. And notice also that, remember that the cells also branch and they can communicate with more than one. Okay, so it's got a lot of similarities and then some differences. And then last but not least, let's talk about smooth muscle tissue. So you would imagine that smooth muscle tissue did not look like this at all because it's not striated, but it's kind of similar still. Here's smooth muscle. Smooth muscle has these little sarcomere looking things. Do you see them at the top that look like actin and myosin? But what happens is they don't come together at Z lines. They come together at these little things called dense bodies. And in addition, they don't line up. Like one of them runs here, one of them slightly that way, and one of them slightly that way. And if they don't line up, do you actually get the stripes? No. You still have that same sliding filament theory of contraction, but you don't end up with stripes. And since they're not all running just parallel to one another, all along one axis, when they contract and they're all running at different directions, you not only make it shorter, you also squish it, right? So you squish the cell. So here's the relaxed one and here's the contracted one. So you squish the whole cell and sort of change it. That's good for the wall of a hollow organ, right? When you contract it, you actually squish it. So that's how smooth muscle is different. It's also involuntary. The cells are much smaller. Now, how would this end up causing things to move through tubes? And I think we talked about this with you guys before, but we'll do it one more time. So if you took a hollow tube, again, like the small intestine, and you wanted to move something through the small intestine, if you had 2 layers of smooth muscle, you could get it to move through by peristaltic contraction. So here's my small intestine. I have an inner layer closest to the lumen that is arranged circularly, yeah? And I've got an outer layer that's arranged longitudinally. And what I'm going to do is I'm going to alternate contraction of those two. And when I squish the inner layer, what happens to the diameter of the lumen? It decreases, right? And when I pull, what happens is I pull stuff through. So if I want something to move, and I do, from proximal to distal in my digestive tract, right? Because you don't want any poop in your mouth, right? So you want it to move from proximal to distal. You squish it, and then you start a pull proximal and go distal. And since the cells are usually connected via gap junctions, if you start at proximal, it'll go distal all the way down, and then you start at proximal again. So you squish and pull, and squish and pull, and squish and pull, and stuff keeps moving, right? That's called peristalsis or peristaltic contraction. If you are moving solids like in the stomach, a lot of times you'll have another layer, 3 layers instead of 2, right? Does that make sense? So you just have to a little bit more musculature to move it more than to move things that are bigger. That's how smooth muscle contraction works. It still starts with actin and myosin though. Okay, I will see you guys on Wednesday.